All right. I hope everybody is doing well. One announcement, particularly if you if you've been paying attention to the YouTube stuff. Apparently, my other microphone died on Wednesdays. Only on Wednesdays, it would die halfway through class. And I was smart on this past Wednesday in that I set up a backup. So I had recording, although you will see the recording changes dramatically as I switch to the backup mid-lecture. So I have a new one. So hopefully this one does not die, or at least doesn't die on Fridays. And we're good today. I did not finish grading your exams. I'm working on it. I'm in the way through, but it took me longer to do other stuff this morning than I expected. So I'll have them for you when you get back from break, because I was trying for today. So I'm on the part with all the flow cytometry and the B-cell development part. Just a couple things to mention really briefly. Remember that with antibodies and B-cells, the antigen can be anything. The antigen can be protein, lipid, nucleic acid, carbohydrate. There really is no way to tell which one it's going to necessarily be. And that's a compare and contrast with T cells, and so that's why it's kind of important that we think about this now. Other thing to be aware of is I asked you, and this came to me as an idea a couple of years ago, when we're thinking about RAG, I asked you which cells have the DNA and which cells transcribe and translate, which is just another way of saying express. All cells have the RAG DNA. Only pro and pre-B cells do the expression, do transcribe and translate. Some of you guys wrote about immature because of receptor editing, which just totally makes sense. It's totally fine. But note that all cells, even the skin cells, even the non-B cells, have the DNA. I was kind of surprised that some people wrote certain types of cells have the DNA and other types of cells transcribe and translate. And I'm like, well, then how can they transcribe and translate if they don't have the DNA? So I was a little confused about that. So be aware of that as you're thinking about stuff. That when you see questions like that, that's kind of something to be aware of. And one reason why that is actually relevant to what we're going to talk about today is that I have in the past gotten questions from people about when we talk about MHC. Because today, for example, we're going to start by talking about some details of the MHC genetics. And then we're going to talk about the MHC proteins. And I sometimes have people say, I'm confused, is MHC a gene or a protein? And I say, it's a gene that encodes a protein. And so it's the same kind of thing. There is a RAG gene that encodes a RAG enzyme or a RAG protein. And so sometimes we do talk about the DNA, the gene, and sometimes we talk about the protein. Today we're going to see both for MHC, and I know that that's been something that sort of confused people in the past. As you see here, we're talking about MHC structure and function. And the first bit that I have to tell you about is related to some MHC genetics and sort of nomenclature stuff. We talked last time about the fact that the MHC genes are the most polymorphic genes in the human genome, meaning there are the largest number of alleles, the most number of different versions of the genes out of any gene in the human genome. And so the reason why this is where we're starting is that we have to think about the fact that we have to think about what do you call all those different versions? How do we name the different versions? How do I refer to the type of HLA-A that Emily has from her mom versus the type of HLA-A that Nadia has from her dad? What do we actually call those things? And because we're going to often see some examples in mice, immunologists use mice a lot, we have to sort of talk about what this nomenclature is like in both mice and in humans. I didn't mention this last time, but as you can probably guess, MHC is an acronym. And MHC stands for major, that part's going to matter later, major histocompatibility complex. And the reason for that is that it was partially described in thinking about what tissues could be transplanted among different organisms. So it was about histocompatibility, tissue compatibility. And this was the major histocompatibility complex. When people were studying different cells, it turns out that MHC was also discovered in a separate set of experiments. And I mention this because in that set of experiments, the scientists also came up with a name for the protein they were studying. And they didn't know they were studying the same protein as the MHC people. So there's this other name that's out there as well, because this came from this other area of study. And in this other area of study, they were actually looking at what they thought of at that point as interesting proteins on the surface of white blood cells of moms and fetuses. It was on the white blood cells. It was on the leukocytes in humans. And so they also referred to it as the human leukocyte antigen, or HLA. Now we have MHC, and that's sort of this broad general term. If we are specifically referring to MHC in humans, we often will refer to it as HLA, the HLA genes. And so if you see the name of a gene or a protein that starts with HLA, that is an indicator that we're talking about a human MHC. As you'll see on a later slide, when we talk about mouse MHCs, we refer to them as part of the H2 locus. And so if you see something that's H2 blah blah blah, you should think, oh, mouse MHC. So this is sort of one distinction. There is actually a specialized term for the MHC locus in pretty much every organism. So when I was in grad school, we spent some time with rhesus macaques, and so I would not use HLA or H2 in my experiments. I would use something called MAMU, which is what it's called there. And so there's some weird name for all of them. HLA and H2 are the ones we're going to care about. When we're thinking about the human genes, which are the way that things are shown here, we have our MHC class 1 molecules, HLA-A, HLA-B, and HLA-C, LAC for our class 1s, and HLA-DP, HLA-DQ, HLA-DR as our class 2s. So this is sort of the way that we have those particular genes here. You're seeing versions of those genes here. And the way that we name these different alleles when we're talking about the HLA system is straightforward and sensible and not actually particularly confusing at all. We use a number. So Emily might have HLA-A1 and HLA-A13, and Nadia might have HLA-A2 and HLA-A52, and so on through all of these numbers. And so we just put a number after HLA-A whatever, HLA-B whatever, HLA-C whatever. You will notice in a couple of places you'll see little asterisks in the numbers and sort of subdivisions, because technically things we used to think were like HLA-A1, we realized there were more than one type of that. But just for our purposes, it's just a number. HLA and a number. So it's real straightforward. You can see the HLA locus here that we see in humans. So you can see we've got that class 1 region. It includes A, B, and C genes, as well as some other genes that encode proteins that we'll see later this semester that do some cool stuff. Really, the ones that are in the class 1 region that are shown here are molecules that structurally look like class 1, and they do something that's vaguely class 1 related. But again, they're not our problem right now. You can see the ones in the class 2 region. They've got 2 letters. Again, there are some like DO and DM that are listed here that I haven't listed for you. They are molecules that structurally have some similarities to class 2. They do some class 2-like things. We'll care about DM next week. That's sort of it. And of course, we've got that class 3 region with some other genes of immunologic importance. You can also notice, like if we look at the class 1 region, it actually goes in the order B, the B genes, then the C genes, then the A genes. I am not going to ask you to remember that, but technically it doesn't go ABC on the genome. So you can see all of this here. In mice, people were not doing experiments by looking at a weird protein on moms versus fetuses. In mice, these experiments were done by skin grafting. We're going to see a little bit about skin grafting now, and we'll see skin grafting come back when we talk about transplantation later. So really the idea with skin grafting, it's basically giving little skin transplants. It's basically taking skin from one mouse and putting it on another mouse. And what the researchers determined is if they had mice of one strain, like this orange mouse that we'll call strain B, and they had mouse of another strain, strain K, they could also make some progeny, which are like BK. They have one chromosome coming from the B parent, one chromosome coming from the K parent. So they could sort of look at what was going on with all of these mice. And they could start to try to identify what genes were important for allowing a transplant to be accepted or rejected by passing different pieces of skin between these different mice. And so when they did that, they found things like, you don't have to get super stressed about this right now. Well, there's one place where you might see it on a practice question, something similar to this, but we're going to see again a lot of this with transplantation. You can see that we have this organism, this mouse that has 2 copies of B, the full orange mouse, sort of the purebred orange mouse. It could transplant skin to another orange mouse, another B. It couldn't transplant to a K, a genetically different mouse. And it also could transplant to its progeny, its child, who was half B. You can see that the K parent could transplant to other K organisms, not to B, and could also transplant to the child. And you could see that the child who has B and K could not transplant to either parent, because neither parent has actually both B and K, but could transplant to other siblings. And first of all, some students will take from this, like they get this idea, they're like, oh, I figured it out. This means I can get transplants from my parents really easily. That would be true if you were an inbred mouse. You are not an inbred mouse. And so it's not quite this simple in an outbred species like us. This is definitely an inbred mouse thing. But really the idea was they could tell there was some gene that was involved in this transplantation. And so you had to have the yellow gene to acquire yellow skin. Or you had to have the orange gene to acquire orange skin. Here you had to have both of these genes to acquire the skin. And what these researchers were able to do is do more and more complex genetic crosses to actually narrow down what part of the chromosome was important and to find sort of the gene of interest. Because they were studying skin grafts when they did this, they called this the histocompatibility locus, or the H2 locus. So it was just the histocompatibility locus. They didn't know it was the same thing as the human leukocyte antigen for a while, because they were doing such different types of experiments. The issue with the H2 locus is that some of the naming with the H2 locus is wacky. There's a certain amount where you're like seriously old-fashioned immunologists, you seriously had to pick like the weirdest sounding naming conventions you could. And yeah, I guess they did. And so that's sort of why we have to talk about this, because you will certainly see it in papers and in problems. And so just like in the HLA locus, we do have a class 1, a class 2, and a class 3 region. The class 1 genes are labeled in red. The class 2s are labeled in blue. The class 3s are labeled in green. You will also notice that in this situation, the class 1 locus is divided. Again, I'm never going to ask you to draw that. I'm never going to ask that you can draw out where the divisions are. But yes, if you are looking at this, that you do see a division. And in mice, we have some class 1 genes, just like we have in human. In mice, our class 1 genes are called H2K, H2D, and H2L. Again, it's one letter, KDL. And in mice, the class 2 genes are H2IA and H2IE. So again, class 2's 2 letters, class 1's 1 letter, just like we saw last time. And so here you can see K, D, and L, and you can say this is part of IA, this is part of IE. So we've got class 1s, class 2s, they still kind of hit the same kind of nomenclature stuff. The thing that is super annoying about the H2 locus is the way that we label different versions of K, or different versions of D, or different versions of L. If you remember with HLA-A, with A, B, and C, we labeled them with numbers, right? It'd be really nice if they used numbers in the mouse system, but they don't. Instead, they do something a little bit different. When they were coming up with the names and sort of trying to describe what's going on in this process, they had a bunch of different mouse strains that they had discovered. I show you 2 mouse strains up at the top, which are the 2 that immunologists use most frequently. One is called the Biol mouse. It's the white one with the red eyes. You can also see the C57 black 6 mouse, also known as black 6 or B6, up in the upper right. But they had a bunch of different mouse strains, including Biol and black 6. And they looked at exactly which kind of K those mice had. They looked at which kind of D. They looked at which kind of L. Because these were inbred mice, they would have 2 of the same Ks. Not like a nice outbred species like us. They have some of the same worries as the cheetah, because they've got 2 of the same. They got the 2 of the same K. They got 2 of the same D, 2 of the same L. And instead of giving them numbers, K number 1, D number 1, they picked a letter for each mouse strain. And so, for example, in a Biol mouse, they referred to all of the versions, all of the different MHCs as the D version. So they said that the IA that is in a BALB/c mouse is called IA of D. And they said that this is IE of D. And they called this one K of D. And they called this one D of D. And they called this one L of D. And so they use these weird little superscript number or letters. And in the case of BALB/c, they're all D. And in fact, in the BALB/c mouse, because it's an inbred mouse strain, it's got a chromosome from mom with the D versions and a chromosome from dad with the D versions. So you would have something that looks like this. In the case of a black 6 mouse, they called all the versions of all the things the B version. I remember that one for B is for black. So in a B6, we have IA of B, IE of B, K of B, D of B, L of B, IA of B, IE of B, K of B, D of B, L of B. So I would see this in a black 6 mouse. And they also had a name that they would use for the entire chromosome sometimes. So if I look at this chromosome that's coming from BALB/c, every single gene, every single MHC gene there is the D version, right? And so I could actually call this the an H2D chromosome. And so if I ever told you, oh, this mouse has H2D chromosome, that means you could expand it out, draw the whole chromosome, and that means you should put a D next to all of them. That's like a shorthand to tell you to represent this whole thing of the D chromosome. This one would be H2B. And so there are some mice, like here you can see, the CBA mouse has K for everything. That was just how it was defined by some person back in the day. This one has D of everything. This one has B of everything. There are also some that weren't the mice that they used for these original studies. And now when they've gone back and looked at them, they're like, oh, they have they have a K that's the same as the K from a BALB/c mouse. And they have a D that's the same as a black 6 mouse. So they might have, for example, in this organism, or in this mouse, it has K of B, it has IA of B, but it has IE of K and D of D. So basically these subscript letters are telling you the versions, just like you see with the numbers. So if I were to go into New York City and I was going to catch a mouse in the subway, I could look at that mouse's chromosome. It would have two copies of this chromosome, one from mom, one from dad. And it would just have random versions of all of them. It wouldn't be like a perfectly inbred strain like in the lab. So it might have the same IE as a BALB/c mouse from its mom. So that would be IA of D. It might have the one that came from a black 6 mouse from its dad. So that would be IA of B. Maybe it has a weird one, IE of K, and then it has IE of D. And then maybe here it's K of B and K of D. Maybe we'll get a D of D and a D of B. I'm just not feeling very creative with my letters. But so the idea is that if you're actually looking at like a wild outbred organism, you just have different superscript letters telling you which version of these genes you have. And that's instead of the numbers that we're using in humans. And we wouldn't have a shorthand like this for that mouse from the subway, because it's got all sorts of variations in which versions of the alleles it has. And so this is sort of what we often think about with the H2 locus. For reasons I do not entirely understand, I kind of do understand them, actually. The slides I'm going to next, I did not put next, but I'm going to be skipping around a little bit on where we are with the slides. But to go to the place where I want to go in the slides, I have to introduce one other word to you. And so that other word that I want to tell you about, it's actually here, it's in a few different places you'll see around MHC, is this word haplotype. And so you can see this word haplotype. And a lot of times people talk about MHC haplotypes. A haplotype is a group of genes that are inherited together. So all the MHCs that you got from your mom is your MHC haplotype. It's one of your haplotypes, actually. You have two MHC haplotypes. The group you got from your mom and the group that you got from your dad. And so you have these two MHC haplotypes. And in fact, most likely, not 100%, but most likely, the MHC haplotypes you have are like exactly, will be exactly one of the chromosomes your mom had. So they'll be like an exact chromosome that she got from her mom or her dad. And you can sort of watch this same chromosome moving through evolution. And moving through generations. There's technically a haplotype, like I said, which we're using as a group of genes that gets inherited together. We often, in hardcore genetics, we talk about as a group of genes that are inherited together without much recombination. So we don't tend to see mixing and matching a lot. It's sort of like those genes always travel together. We don't seem to see a whole bunch of mixing and matching. The MHC region is actually known as the longest haplotype in the human genome. So you tend to see sort of that section in the from your mom to you. You don't tend to see much recombination to get mixing and matching. And just to give you an understanding of that idea of a haplotype of genes being inherited together, I said that the MHC locus is the longest haplotype in the human genome. The second longest haplotype in the human genome is a section of a chromosome, I don't remember which chromosome number it is, that has eye color, skin color, and hair color genes. And so if you think about it, there's sort of this stereotypical, like blonde goes with really pale skin and blue eyes, right? You often see those traits together, but it's not 100%. It's not always that they're coming together. And so that's the second longest haplotype, or set of genes that are usually inherited together, because we don't see a lot of recombination between them. And so the reason why I'm telling you all of this, and the way that I'm going to switch forward in the slides, is I actually have two different little ways we can look at using this haplotype information in a couple of problems. Because thinking about MHC haplotypes and genetic typing is something that people do a lot when we're trying to really do typing of some sample. If we're in a forensic situation and we're trying to say, like, is the DNA at the crime scene the same DNA as the killer? What we usually do is check what the MHC combo is. And then we try to see, is the MHC combo of the killer the same as the MHC combo at the crime? And so we'll say, there's a this many chance probability that it's the same. It's because it's like how many of the MHCs are the same. And so we often do that. Also, when somebody does something like paternity testing, it's entirely done by MHCs. And so this is an example from a textbook where we have an offspring we have a biological mother, and we have 3 potential biological fathers. And we can look at the MHC of these individuals and try to figure out which of these fathers is the potential father. And so what do you think from this? And then I'm going to show you a way that I've often asked it have often asked similar things on an exam. So you can be used to the nomenclature I use. Yep, Emily.
Potential father 2.
Potential father 2. Why potential father 2?
Because, well, I actually did look at B or C, but
Okay.
A, it got A3 from its mother, and then
Mm-hmm.
it would have gotten 43 from its father, and potential father 2 had 43.
Yeah, so we can see the offspring has A3 and A43. Mom has A3 and A11. It must have gotten A3 from mom. So it must have gotten A43 from dad. The only one that has A43 that it could have given it to them was father number 2. And we can check with our B and our C. We can see that mom gave 59. Or sorry, no. Yeah, mom gave 59 and dad gave 54. And for C, mom gave 8, dad gave 5. And so we can see that it matches, it works out, that potential father 2 is the father in all 3 of these ways. So the way that I have asked this in the past, I did once ask a thing like that, where I actually used Mamma Mia, because I was watching Mamma Mia while I was reading, while I was writing the exam, so we had to figure out who in Mamma Mia was the father. But here, what you can see is that sometimes I will show you a father, a mother, and a child, and I will ask you to tell me the MHC haplotypes of the mother and father. And so what I basically want you to tell me in this problem is I want you to tell me each of mom's chromosomes. So for mom, you have 2 answers, 2 haplotypes, which are her 2 chromosomes. For dad, you have 2 answers, which are the 2 haplotypes or the 2 chromosomes. You can also see this is slide 38 in these slides. The previous one was slide 37. And so if we were to try to answer this, what would be the answer? So we need to have 2 haplotypes for mom and 2 haplotypes for dad. And remember, a haplotype is a group of genes that gets inherited together. So I'll do one of the 4. I also sometimes think about this as the chromosomes. So mom's 2 chromosomes, dad's 2 chromosomes. Well, dad has HLA-A1 and HLA-A20. One's on one chromosome, one's on the other. We'll just say A1 is on one of them, A20 is on the other. Okay. Dad clearly gave A20 to the child. That was the only thing that could have happened. Okay? So now let's look at dad and the child and B. Well, dad gave the child B57. What that means is that B57 must be on the same chromosome as A20 so that they could be transferred together to the child. And that means that the other B, B7, must be on the other chromosome, dad's other chromosome, which is not the one he gave to the child. We can also look at, I gave you DR in this case for dad. Dad must have given this child DR5. So you got DR5 on this chromosome that dad gave to the child. And DR2 must have been on his other chromosome. So sometimes I just write out the 3 that they gave to the child, and then I just write the other 3 as the other chromosome, the ones that are sort of left over by process of elimination. So what are mom's 2 haplotypes? Yeah, Jazz. You can give me one, absolutely.
So the child has A2 and A20, but
Mm-hmm.
he got A20 from the father, so he got A2 from the mother.
Okay, so we got A2.
And then B27 must be on the same chromosome.
Okay.
And then the mother has 2 DR3s, so obviously on on that one chromosome, it must be DR3.
All right, so one of mom's chromosomes is A2, B27, DR3. What's mom's other chromosome? Aurora.
A3, G12, DR3.
Exactly. So basically the idea is we're listing out each chromosome of the parents. So this is when it asks for identify the MHC haplotypes of the mother and father in this family. This is what you would do. You can see this question was on this previous, whatever it came from, exam problem set, I don't know what, was worth 4 points, and there's 4 answers. So it's like 1 point per answer. So that's how you would do a question like that. So that's sort of my general information about MHC genetics. But now we need to think a little bit about some details of the MHC protein. So we're going to be thinking about some specific details of what this protein actually looks like, and some aspects of its structure, and some other key things. So remember that we've already seen the fact that MHC is this cell surface protein. It has this pocket that is binding some part of a peptide from a microbe. And so this is different than when we had B cells, because here it's only proteins that are the potential antigen, not all 4 different types of biomolecules. The part of this process where we degrade the protein and put it into MHC is the part that we're talking about next week. Right now, what I want to talk about is sort of what this protein that's on the surface looks like, structure-wise. In order to do that, I have to give you a brief biochem review. So, this is how amino acids come together to make proteins. You can see that we have our amino acid with a nitrogen on one end, that's the N-terminus or the amino terminus. You can see that we have the C double bond with an O, and then either an OH or an O negative, that's the carboxy terminus or the C terminus. So those are like the ends, the beginning and the end of the amino acid. We've got a carbon in the middle that you can see, and we have on one side of that carbon a hydrogen and something else on one side, an R group. And the R group is where different amino acids are different from one another. And so I'm going to mention R groups a few times. So that's the R group. And we'll see some more about R groups on the next slide. When we make a protein, you can see that we link together our amino acids like this. So here you can see one amino acid. There's its amino terminus, there's its carboxy terminus, and that carboxy terminus links to the amino terminus of this next amino acid. And then here's a carboxy terminus, and it could go on and on and on in a super long chain. And so you can see we sort of have NCC, NCC, and it could go NCC on for a long time. That is something I'm going to reference as the peptide backbone, or the backbone of the protein. So I'm sometimes going to mention the backbone, and I'm sometimes going to mention the R groups. And so you can see both the backbone and the R groups in looking at this. And so here you can see a little protein here where we've got our one amino acid with its R group, another one with its R group, another one with its R group. And you can see those R groups differ. And we also have our backbone of NCC, 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 NCC. Okay, so when I say backbone, that's what I mean. When I say R group, that's what I mean. And we've got 20 different amino acids. These are their R groups. I'm not going to ask you to remember all of the R groups right now. But there are a couple things I want to point out about R groups, and just a couple other little amino acid related things that I want to point out because immunologists often use them, or because you will see them come up frequently as I talk as in examples. So you can see we've got a lot of different sort of chemical moieties as R groups. In general, the thing that we care a lot about with those R groups is the fact that they differ in size. So there's some that are really small, like lysine. Look, it's just a hydrogen. And there's some that are really big. Look at this giant thing for tryptophan. So they vary a lot in size. They also vary in things like charge. So some of them have no charge. Some of them have a positive charge. Some of them have a negative charge. Some of them are sort of like wide and bulky. Some of them are long and skinny. And so we're going to see the idea that there are going to be some binding pockets that are going to be specific for only some R groups. Maybe they care about whether the R group is a plus or positive or negative charge. Maybe they can only fit a big R group. Maybe they can only fit a small R group. So we're going to be thinking a lot about the sizes of the R groups. Very often, this also has all the names of all the amino acids, as well as their 3-letter abbreviations and their 1-letter abbreviations. Most of the time when immunologists show information about amino acids, they show it in 1-letter abbreviations. And so if I'm ever looking at doing 1-letter abbreviations and you're unsure which amino acid I'm referring to, you have a slide to reference them on. Just so you know, one that everybody in immunology loves to use as their example is tyrosine. Here's tyrosine. Tyrosine is Y. So if I start drawing random Y's somewhere, I mean tyrosine. And tyrosine is kind of interesting because it's sort of big and bulky with this ring, and it has an OH. And so there's some interesting stuff that happens with the OH and with the big bulky. And so tyrosine often is sort of my example. You can see it's pretty similar to phenylalanine. So sometimes people talk about F or Y in certain places because they're relatively similar. But this is sort of why we're caring about R groups. And you can, like I said, see the difference in things like size, shape, and charge. To go any further in telling you about the structure of the MHC protein, we have to start to divide stuff up a little bit. When we talked about the genetics, I told you that there were class 1 genes and class 2 genes in the MHC. It turns out the class 1 genes encode the class 1 proteins. The class 2 genes encode the class 2 proteins. And the structure of the class 1 proteins and the class 2 proteins is different. So I can't just tell you about the structure of MHC because there's really 2 structures. There's the MHC class 1 structure and the MHC class 2 structure. So I'm going to start today telling you about stuff about MHC class 1. After that, we'll talk about MHC class 2. Ooh, that was wrong. And so here you can see MHC class 1 versus MHC class 2. As you're comparing and contrasting, one thing to notice is that MHC class 1 is kind of shown mostly as one color, right? If I was actually going to remake this slide myself, I would make this little oval a different color. And so really, MHC class 1 involves 2 proteins, and they're super different in size. There's this whole big yellow thing, and then there's this other protein. It's only got 1 transmembrane domain, because this other thing doesn't have a transmembrane domain. MHC class 2 has 2 transmembrane domains. You can see it has 2 different proteins making it up. They're shown as different. And you can see that both have a peptide binding cleft. You can also see that some of these domains are depicted as rectangles. Those rectangles are immunoglobulin domains, or the immunoglobulin fold that we saw in antibodies as well. It's the same exact protein fold. I tend not to draw a lot of MHC molecules on the board. If I do, I mostly draw class 1. A, because I spent more of my life thinking about class 1, and B, because I had a very traumatizing experience the first time I was teaching where I drew a class 2 on the board. It did not look like a class 2. And nobody laughed. It was the most phallic drawing I've ever drawn on a chalkboard, and no one in the class laughed. I just kept taking notes. So now I just don't draw class 2 anymore. So we're going to think in detail here about class 1. And again, you can sort of notice this idea with class 1, and that class 1 has this one particular protein, the MHC class 1 protein, that has 3 separate domains, one immunoglobulin domain that also kind of attaches to this transmembrane part, and these 2 other domains at the top. The 2 at the top are called alpha 1 and alpha 2. The one down at the bottom, right by the transmembrane domain, is called alpha 3. And then you can see that MHC class 1 pairs with this other protein called beta 2M, or beta 2 microglobulin. When we talk about variation in MHC and MHC class 1, when I'm drawing these chromosomes with A, B, and C, or K, D, and L as my class 1s, that K, D, and L, or A, B, and C, and all that variation is referring to this sort of larger alpha chain, the yellow chain here. Sometimes it's referred to as the MHC class 1 heavy chain. Beta 2 microglobulin is encoded somewhere else in the genome, and it doesn't vary at all. So basically you have all these different alpha chains, all these different MHC class 1 heavy chains that all pair with the same beta 2 microglobulin. You can see that beta 2 microglobulin does not have a transmembrane domain. You can also see that beta 2 microglobulin is not covalently linked to the rest of the MHC class 1 molecule. And that's actually really important because in reality, we think that beta 2M is actually going on and off of the class 1, and it's sort of unfolding and refolding all the time. We sometimes talk about MHC class 1, how MHC class 1 breathes, because it's sort of going on and off with the beta 2M, and it's not covalently linked, so that can happen biochemically. So we can see our class 1 molecule here. And what you can also notice is that the peptide, which is shown in this image in the right in like orangey, that peptide is bound by the peptide binding cleft. And the peptide binding cleft is made up of the alpha 1 and the alpha 2 domains of that heavy chain protein. So the peptide binding cleft is coming from just that one protein. So the peptide binding cleft is all one protein. One thing that's actually pretty cool is that the alpha 3 you can see is mostly made of beta sheets. The alpha 1 and the alpha 2 both have an alpha helix, and then there's some more beta sheets. And it's like there's like a beta sheet platform and 2 like alpha sheet walls, like bumpers on the side of the cleft. So you've got these 2 little alpha helix bumpers with a little beta sheet tray underneath, making this peptide binding cleft, and then you've got this little beta sheet thing holding it all up. You can see another example of the MHC class 1 peptide binding cleft here. And what I want you to notice is, again, you can see the 2 alpha helices. You can see that they're the same color green here because they're coming from the same protein. And they're going around this peptide. The peptide's binding in between them. Here they're listed as from alpha 1 and alpha 2, but they are the same protein. So you can see here's the little MHC cleft. That's where the peptide would be. And this is actually a space fill model with the white is the MHC class 1. You can see the peptide in the bottom. You can also see a little bit of beta 2M poking out under, but no one cares. And so you can see this example of the MHC class 1 peptide binding cleft. This is, you're the T cell here looking at the MHC plus peptide. And when we look at the MHC class 1 peptide in this way, there's one really key thing that we can notice, and it is especially notable in this space filling model, which is why I use it, but it is something that you can see in all of these models. And the key thing to notice is that these alpha helices that are making up the peptide binding cleft come really close together. And so the ends of this cleft are closed. And so you can see there's stuff here on these by the ends of the peptides. The point is that the MHC class 1 peptide binding cleft is going to be really picky about how long the peptide is. It can't fit peptides of any length because it has this really constrained pocket size. So it can only fit peptides of a very specific length. For MHC class 1, that length is 8, 9, or 10 amino acids. Depends a little bit on which MHC class 1 molecule we're talking about. Each one has a preference. Some will have a preference of 8, some will have a preference of 9, some will have a preference of 10. But the answer is 8, 9, or 10 for MHC class 1. And the reasoning is because the ends of the peptide binding cleft are closed. That really limits the length of that peptide. Yep, Jazz.
But all amino acids have different, as we discussed, different sizes and different charges. So how is it 8, 9, or 10? Like, how is that like a kind of defined amount? Is it based upon the backbone?
That's exactly where we're going.
Okay.
Let's see the next slide and then see if you still have this question. And so the way that this works is that we've got this peptide binding cleft, and it's making biochemical interactions with our groups. And so you can see here is my peptide in the middle of my peptide binding cleft. It's a closed cleft that's only going to have 8, 9, or 10 amino acid preference. And we're going to see a lot of interactions. Here you're actually seeing little biochemical interactions between amino acids in the MHC and amino acids in the peptide. And those are all interactions that are happening with the R group. Another way of looking at this is like this. And so this is what we usually think of when we think of MHC class 1. And so when we think of our MHC class 1, first of all, we have these closed ends. And not only do we have the closed ends, but those closed ends of the pocket even have charges on them. So if we're going to bind our peptide, here's going to be the N terminus, here's going to be the C terminus. An N terminus is often positively charged. A C terminus is often negatively charged, and you can go back to that peptide that I was showing you earlier if you want to talk more about that. And so this end of the cleft actually usually has a bunch of negative charges so that it can make the peptide go in the right direction. This side has a bunch of positive charges to make the peptide go in the right direction, and we have it sort of stretched out in a line. Some R groups are going to be pointed up towards the T cell. Some R groups are going to be pointed down. Does that make sense, Jazz? So in some of those R groups, they're going to be pointed up, and that's going to be what the T cell receptor is recognizing. Some of them are going to be pointed down, and that's going to make the peptide lock into the pocket. And that's going to make this particular MHC class 1 specific for certain kinds of peptides. And so we're going to have, you can see some of these R groups are going to sort of be making contact with the T cell. They're going to be sort of T cell contact residues. But we're also going to have some that are going to have a specific shape. So here one's kind of roundy shape, and here one's kind of squarey shape. We're going to have these sort of specially shaped like little dips in the pocket. I was going to say pockets in the pocket, and that doesn't make sense. That are going to bind only specific kinds of R groups. And so only a peptide with that amino acid in that position will fit into this this particular kind of MHC class 1. And we refer to those amino acids and those R groups as anchor residues. And so with MHC class 1, we see this phenomenon of peptides having specific anchor residues that bind them into the peptide binding cleft. And so you can see this here as well as on the next couple of slides. So here, somebody actually purified 2 different kinds of MHC class 1s. So they actually took cells, cut the MHC class 1s off. Maybe one of them was A2 and one of them was B27. I don't know. It just calls them number 1 and number 2 here. And then they actually did biochemical separation to pull all the peptides out. And they looked at the peptides. And they found that MHC type 1 could bind any peptide that was 9 amino acids long, that had a G at position 2, a P at position 3, and an I or an L at position 9. And MHC type 2 could bind peptides that were 9 amino acids long, that had a Y at position 2, and an I or a V at position 1. And so that was based on where the pocket was set up in MHC type 1 or MHC type 2, like where the part for the R group binding was, and what the size and the shape and the charge of it was, in terms of what R groups it would accommodate. And so for MHC type 1, we would say that the anchor residues are a G at number 2, a P at number 3, and an I or an L at number 9. For number 2, we would see the anchor residues are a Y at position 2 and an I or a V at position 9. So this sort of gets at this idea of the anchor residue. One reason why a lot of students often like this experiment, you can see this experiment is like textbook experiment, and you can see it really helps with your understanding, right? This experiment was an honors thesis project by 2 undergraduates in Germany. And so, you know, people do legit research as undergrads. Yay, go undergrads. And so this is sort of, so each of those peptides would be referred to as an epitope that's being presented to a T cell. And those MHC class 1 epitopes have anchor residues. You can see this in this image as well. So you can see that, you know, we have our peptide of some length, and for each individual MHC class 1 molecule, there's some pattern of what you always see. So like in this one, we always have an L or an M at position 2, a V at position 6, and a V or an L at position 9, and then it gives us an example peptide. And then here's a different MHC class 1 molecule. It wants an R at position 2 and an R or a K at position 9, and here's an example peptide. And at the bottom, and you've got 2 different mouse MHCs, an example of K of B versus an example of K of D. And this shows you a bunch of different peptides that they can bind. And so you can see, you know, where the anchor residues are and what the preferences are. And you can also notice that, say, K of D can bind to peptides from many different microbes, not just one. So this particular combo of amino acids might be present in multiple different peptides from multiple different microbes. So there are a couple other key things we need to know about MHC class 1. One of the other big distinctions between MHC class 1 and MHC class 2 is how and where they get their peptides. And that's really what we're going to be talking a lot about next week, is how and where they're getting their peptides. But the big picture answer for class 1 is that MHC class 1 is getting its peptides from the cytoplasm of the cell. So MHC class 1 is basically able to get peptides out of the cytoplasm of whatever cell and show them off on the surface of the cell. We usually refer to that as presentation. So MHC class 1 presents cytoplasmic peptides. If you were to think about this, at least kind of simply, and it's also the way it's shown in this picture, because it's the way most immunologists think of this. If you think about this, what kind of microbe very likely has its peptides just hanging around the cytoplasm of a cell? Yeah, Emily. A virus. So usually the viral peptides are going to be in the cytoplasm. You can imagine like a bacteria, it might be in a phagosome instead of just floating free in the cytoplasm. Virus peptides are often going to be in the cytoplasm. And so oftentimes we think about MHC class 1 as being really important for presenting viral peptides. It's not 100%. I can give you definite exceptions to that rule. But to be honest with you, if I give you some question and I'm like, there is this virus, what's going on with its MHC? I want you to say, if it's a virus, that it's going on class 1. If it's a virus, I'm trying to be like, ding, 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 class 1. And so it's very often class 1 and a virus. So now let's think about it. We're presenting a peptide on MHC class 1 that has come from the cytoplasm, very likely from a virus. And T cell sees that on its other cell, on this other cell that's presenting with the MHC. What's the best thing to do about that virally infected cell? What can you do about that cell that's infected with a virus if you want to be a good immune system? What should you do? What do you think? You should kill that cell. You're not going to like save that cell. If you find that cell and it's like, here, I got this thing, it's from a virus, you should kill that cell, right? Remember that. It's going to come back in a second. If we actually look at sort of some biochemical interactions, and you try to purify the T cell receptor binding to MHC plus peptide, you actually find out that binding is relatively weak. You don't need to worry about all the details of it, but just notice T cell receptor binding is weak in terms of biochemical binding. Other kinds of receptors bind much stronger, but T cell receptor binding is weak. As a result, T cells really need a lot of additional proteins to help make interactions happen. Just the T cell receptor MHC interaction's not enough. It's kind of where we get the specificity from, but we need to have an awful lot of other proteins here helping out to hold the two cells together, because MHC plus peptide is just not strong enough. And what I want to mention here is what one of those other proteins is that really contributes a lot to helping stabilize this interaction. This is going to be a protein on the T cell that is helping the T cell bind to MHC. It's helping the T cell bind MHC better. It's basically, the T cell is going to grab on to MHC, and this other protein is going to also grab on. And so we're going to have two things grabbing onto this other cell and binding it. When we talked about T cells, I told you there were broadly two kinds of T cells. And those two kinds of T cells were either CD4 T cells or CD8 T cells. CD4 T cells had this protein CD4 on their surface. CD4s had CD4, CD8s had CD8. CD8 is specifically the protein that is used to stabilize an interaction with MHC class 1. So MHC class 1, you can see here, presents its peptide, and that MHC class 1 binds with the T cell receptor, but it also binds with CD8. And so CD8, you can see, sort of is reaching and binding to like the back part of the MHC, a part that's conserved between all the MHCs. So MHC class 1, when it's showing off peptides, it's not actually going to be able to show that peptide off to any old T cell. It's only showing off that T cell, that peptide, to a CD8 T cell. So MHC class 1 specifically interacts with TCR and CD8. When people think about trying to remember this later, they often think about, you multiply the number of the MHC and you multiply the thing on the T cell together, So here you have 1 times 8 gives you 8, right? In the other one, it's going to be 2 and 4. So you always have to get 8. It's either 1 times 8 or 2 times 4. So MHC class 1 is specifically the kind of MHC that turns on CD8s. And as Jazz pointed out to us, what do we want to do if we have a peptide on class 1 that came from the cytoplasm? What do we want to do to that infected cell? We want to kill it, right? What do CD8 T cells do? Kill! It all fits together. There's one other piece. Okay, so MHC class 1 plus peptide is the antigen for CD8 T cells. So you can see we've got cytosolic peptides binding to class 1. That turns on a CD8 positive T cell. So we can see kind of all of these pieces coming together. There's one other piece that we have to put together for MHC class 1, which is what cells are doing this process. And again, we're going to sort of be simplifying here and say class 1 is usually presenting viral peptides. So if you think about all the cell types in the body, which cell types in the body could get a virus infecting them? So Brooke says all of them. Does anyone have any asterisks they want to put on that? So it's actually all minus 1. Is that the actual answer is all minus 1? Do we know what does anybody have any idea what the 1 is? Not bone marrow. So the 1 is red blood cells. Red blood cells don't have a nucleus. So what the heck's a virus going to do to them if they have no nucleus and they're not doing any like transcription, translation, replication stuff? So sometimes we talk about nucleated cells, and nucleated cells are everybody minus red blood cells, because red blood cells don't have a nucleus. And so we can look and say which cells express, again, this is transcribe and translate, MHC class 1. And the answer is all nucleated cells. So basically, it's a yes for everything but red blood cells. Red blood cells are a no. Everybody else makes MHC class 1. In fact, every cell of your body is making MHC class 1 24/7. So every cell of your body right now, 24/7, really does have 2 A's, 2 B's, 2 C's on its surface showing stuff off. 24/7, unless it's a red blood cell, where we're going to see this codominant. So that's where we see this codominant expression. And so that's our information about class 1. I'm not going to have time to get through all of class 2 right now. So I'm only going to tell you a little bit of stuff about class 2. And we'll finish off with class 2 next time. So if we think about MHC class 2, it does look rather different than MHC class 1. In particular, it has 2 chains that are basically equivalent in size, known as the alpha chain and the beta chain. So when I drew my chromosomes, I was just drawing IA and IE. Really, there's 2 IAs and 2 IEs, one for the alpha and one for the beta chain. So in fact, MHC class 2 is made up of 2 equally sized proteins. They both vary. This isn't like beta 2M, but it's like its own separate thing. Both of these chains have a transmembrane domain. And the 2 of them come together to make the peptide binding cleft. So again, you see our peptide binding cleft. It's got 2 alpha helices putting together that and the peptide in the middle, but this is coming from actually 2 separate proteins, the alpha and the beta chain of class 2, each of whom has its own transmembrane domain. And this is actually super important. So one of the key things about class 2, when we think of the peptide binding cleft is that if we think about the edges or the ends of those alpha helices in the peptide binding cleft, it turns out that they're not open. Or sorry, they're not closed. They are open. They're open ends. And so you can also notice there's not really anything like around the ends of the peptide. And so this shows the peptide is like the same size as the peptide binding cleft. But in fact, the peptide can like hang out of the peptide binding cleft. It can be kind of any old length it wants and like flop out the ends. This is where some immunology textbooks show a hot dog in a hot dog bun. But the idea is this peptide can kind of be any length and be flopping out the end with class 2. We don't have a strict length. Sometimes people talk about like, I don't know, 14 to 20, but it honestly can be any length. I've heard of like crazy long peptides. And so it's really any old length we want it to be. In addition, if you actually pull those peptides out and you try to look at their amino acid sequences, you find you really don't see like anchor residues. You don't see like particular amino acids being important here. In fact, from what we can see, MHC class 2 is making all of its interactions with the backbone, or the main, which is, I know the title of this says main chain, but I was telling you earlier today, backbone. So that NCC, 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 NCC that doesn't look different no matter which protein it is, that's largely what we're seeing interactions with in class 2, not the R groups. And so MHC class 2 is not nearly as picky as class 1. It can kind of take a peptide of any old length, and it doesn't really care about amino acids. So every so often I'll see a paper where someone will try to tell me about a class 2 anchor residue, but really there aren't class 2 anchor residues. Because like, look at this. Here it's number 5, and here it's number 4, 5, 4, 4, 7. Like, it's not the same number, and it's really not the same amino acid. Here again, really not the same amino acid, and really different numbers in the protein. And so you really can't pick out an anchor residue. You can't like look at a protein and say, aha, this is the part that's going to bind class 2. It's not really something you can predict. One other thing I want to tell you about structurally for class 2, and we'll do the rest of the class 2 stuff later, actually comes up again here, and is going to be something we're going to care a lot about next week. So, I know a few of you do Western blots in your research lab. And when you do your Western blot, you have your sample, and you put it in your SDS blue buffer, put it in your loading buffer, and you put some beta-mercaptoethanol in it, or some DTT, and you boil it, right? Why do you do all that? What's the point of all that? Yeah, Emily?
To get the proteins into their primary structure.
So it's to denature them. So it's to unfold them. If you have proteins bound together in complex, it pulls them apart. So you have individual proteins pulled apart and made into a line, right? Because SDS is really good at breaking a lot of types of bonds, right? To help linearize that protein. MHC class 2, when it has peptide, is really strong. Really, really, really strong. It's actually known as an SDS stable complex. And so if you put this with SDS and you boiled it, it would not come apart. Once you have MHC class, the two parts of your MHC class 2 molecule and a peptide, it's really, really, really hard to take that apart. And so the things that we normally do to sort of pull proteins apart in biochemistry don't work on MHC class 2 plus peptide because it's so very stable. With class 1, we have this other protein that's not covalently linked and doesn't have a transmembrane domain, and so it can kind of come on and off and on and off. Here, it is crazy, crazy, crazy stable when we have this whole complex together of our class 1, or our class 2 and our peptides. This will be relevant, I'll say it more again next time, but just also know that in neither case, class 1 or class 2, in neither situation will it fold correctly until you have the peptide. So you don't get everything folded exactly right until you have a peptide in the peptide binding cleft. You need the peptide for proper folding. And we'll see that more next time. So have a great break. You'll see some emails from me about some stuff coming up after break. And yeah, have a good time.